Good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. Yesterday we have begun with plant physiology and we are continued with the early discoveries and the photosynthetic pigment. Clearly we have done or discussed with action spectra and absorption spectra. Today we are clearly going to study about chloroplast structure, photosynthetic pigments once again, and we are going to clearly look into the locations, reactions that are happening, three sequential reactions of light reaction, and we are clearly going to learn the cyclic photophosphorylation and non-cyclic photophosphorylation, which is most important and crucial for our exam. Before getting into the concept, let me give you a brief introduction about NEET World's special programs. We have NEET World's 2 steps to medicine with the minimal fee of 3333 per month, and we have NEET World 1 step to medicine and NEET World's power step to medicine. 3 of the special programs are having the same fee of 3333 per month, in which the complete lecture content of the entire syllabus NEET 2026 is dealt, and along with that we are also going to give you the complete study material and 2 master revision strokes both in the online and offline mode. For more details, contact the given number or you can directly visit our branch, which is in Narayanguda, Hyderabad. So yes, let's begin with the chapter photosynthesis in higher plants. First, today let us learn about the chloroplast structure. Let's begin with the chloroplast structure. This chloroplast structure, the questions will be asked connecting the double membrane bound organelles of cell, the unit of life, and photosynthesis in higher plants. Both the areas we are learning the organelle chloroplast, right beta? So we know that chloroplast is a double membrane bound organelle. It has outer membrane and inner membrane, and inside fluid filled space or this matrix, this fluid filled matrix, inside this chloroplast is called as what ma? Stroma. This is said to be stroma. And inside this fluid filled matrix, that is stroma, the sac like structures which are present are called as thylakoids. The sac like structures are called as what beta? Thylakoids. These thylakoids have a membrane, so we call it as what? Membranous sac like structures which are present in the chloroplast are called as thylakoids, and thylakoid membrane is called as thylakoid lamellae, and this is said to be the third membrane of the chloroplast also. Okay, this is the third membrane of the chloroplast, thylakoid lamellae or thylakoid membrane. And the stalk of sac like structures is called as what? Grana. Stalk of sac like structures is called as grana. Clear, right ma? So here the chloroplast structure, it is double membrane bound, and inside the fluid filled space is called as what? Stroma, inside which you have membranous sac like structures called as thylakoids. Stalk of sac like structure is called as what? Grana. And inside this fluid filled matrix, what do you find inside the stroma? In stroma you find the starch granules, lipid droplets, ribosomes which are 70S ribosomes, okay, and then you can also see double stranded circular DNA. So amongst this constituents, ribosomes which are 70S and double stranded circular DNA, these two you can see in a prokaryotic cell, and then you can see in a eukaryotic cells chloroplast and mitochondria only. You can see both of them. What are they beta? Ribosomes which are 70S and double stranded circular DNA. Both of them are present in chloroplast and mitochondria. Why is it so? Because these two organelles have evolved from the prokaryotic cells, which we learn it under what? Endosymbiotic theory. Okay, endosymbiotic theory. Fine. In endosymbiotic theory, it is very evident that chloroplast and mitochondria are once upon a time, they are prokaryotic cells only, but in due course of evolution, they have entered into the eukaryotic cells and they have some prokaryotic features like what? Ribosomes, 70S ribosomes and double-stranded circular DNA. And we all know that the ultimate goal of DNA is to synthesize the protein, right ma? The ultimate goal of DNA is to synthesize protein. And this double-stranded circular DNA which is present in the chloroplast will synthesize world's abundant protein, that is rubisco. Okay, rubisco, which is the world's abundant protein, is synthesized by what? Double-stranded circular DNA which is present in the chloroplast stroma. This is the complete concept with regarding to the chloroplast structure. Let us once recall whatever we have learnt visualizing the NCERT diagram, that we have what? Double-stranded circular DNA, we have both outer membrane and inner membrane, and inside this fluid filled matrix is called as what ma? Stroma, and inside stroma we have starch granules, yes we have starch granules, lipid droplets, 70S ribosomes and double-stranded circular DNA which will synthesize the world's abundant protein, that is rubisco. Fine ma. And sac-like structures are called as what? Thylakoid. Stalk of sac-like structures are grana. One grana is connected to another grana by what? Stroma lamellae. One grana is connected to another grana by what? Stroma lamellae. So now we have got the complete idea, right ma, regarding the chloroplast structure. Now let us look into the light harvesting complexes which are present in the thylakoid membrane. So over the thylakoid membrane, if I have to assume this as thylakoid membrane, just a second. Yeah, let us consider this as thylakoid membrane. In this thylakoid membrane, we will be having photosystems. Photosystems. Fine. As such, in higher plants like in the thylakoid membrane, we have two photosystems, PS2 and PS1. Each photosystems will be having what? Light harvesting complexes. Each photosystem is having what? Light harvesting complexes. Light harvesting complexes will constitute two type of pigments. Those are called as accessory or antenna pigments. Antenna pigments. Fine. These are called as accessory or antenna pigments and core pigments. Fine. So we have in light harvesting complex, we have two types of pigments, accessory or antenna pigments, and we have core pigments. This accessory or antenna pigments will include like carotenoids, xanthophylls, phycobilins and chlorophyll B. Accessory or antenna pigments include what ma? Carotenoids. They include carotenoids, xanthophylls and chlorophyll B. Fine. So accessory or antenna pigments include carotenoids, xanthophylls, chlorophyll B. They absorb light at lower wavelength when compared to chlorophyll A and at different wavelengths. Okay, and after absorbing that light in the form of photons, this accessory pigments or antenna pigments, they will submit it to the primary acceptor or reaction center, which is called as chlorophyll A. Okay, so here chlorophyll A is what ma? Reaction center. Reaction center or primary acceptor. Primary acceptor. Clear, right beta? So here we have considered this as what? Thylakoid membrane, and inside the thylakoid membrane we have two photosystems. Inside each photosystems we are having light harvesting complexes, is that clear? And light harvesting complexes are having what? Accessory or antenna pigments and the core pigments. This accessory or antenna pigments include what type of pigments? Like carotenoids, xanthophylls and chlorophyll B, and this core pigment is including what? Chlorophyll A, which is the reaction center. Antenna or accessory pigments will absorb the light at different wavelengths and they will submit that energy to what? The chlorophyll A, which is the reaction center. Clear, right ma? So complete light harvesting complexes are present in photosystem 1 and photosystem 2. The wavelength of reaction center, that is chlorophyll A, maximum wavelength of photosystem 2 will be 680 nanometers. Okay, reaction center maximum wavelength is how much ma? 680 nanometers, and photosystem 1 chlorophyll A maximum wavelength will be how much? 700 nanometers. Clear, right beta? Next. Now let us look into the sequential steps that are happening in photosynthesis. As we all know, from the standard equation of photosynthesis, the primary product of photosynthesis is what ma? Glucose is the primary product of photosynthesis and oxygen is evolved as a byproduct of photosynthesis. This we all know. Okay, sunlight in the form of photons and happens because of what pigment? Chlorophyll pigment. So the ultimate goal is what? To form the primary product of photosynthesis, that is glucose. As such, here if you can take, okay, first here if you can take what? Photosynthesis sequential reactions. Firstly, we have light reaction and dark reactions. Okay, light reaction and dark reactions. Light reactions are also called as light dependent reactions. Okay, light reactions are also called as light dependent reactions. All these reactions are directly dependent on light, and the further reactions which are continuation after the light reactions which occur are dark reactions. Dark reactions or light independent reactions. Light independent reactions. Calling light independent reactions as dark reactions is a misnomer, because it doesn't mean it takes place at dark, but it only means that they are dependent on the products of light reaction, not directly dependent on the sunlight. Do remember that ma, because we can expect a statement based question asking that light independent reactions is calling light independent reactions as dark reaction is a misnomer. Remember that. Now what are the three sequential events of light reactions? First one is what? Absorption of light in the form of photons. Absorption of light in the form of photons. This is followed by photolysis of water. Photolysis of water, where one water molecule will be photolysed to 2 protons plus 2 electrons plus half O2, that is nothing but one oxygen atom. Third is what? Formation of assimilatory power. Assimilatory power. So this assimilatory power can be what? ATP and NADPH. Fine. So here we have light reactions or light independent reactions. Three sequential events, what are that happening? Photons, okay, absorption of light in the form of photons. Second is what? Photolysis of water, where water molecule is photolysed to 2 protons and electrons and oxygen atom. Third is what? Formation of assimilatory power, that is ATP and NADPH. Now here comes our picture, that is formation of glucose, that happens in dark reaction utilizing this assimilatory power, where carbon dioxide is being fixed to what ma? Glucose, C6H12O6. Clear, right? So utilizing what? ATP and NADPH. The most important segment here we have to focus is what? Locations or sites of the reactions. Light reaction will happen in grana or thylakoid lamellae. Grana we will call it as, more specifically what ma? Thylakoid lamellae or thylakoid membrane. Fine. So light reactions occur in grana or thylakoid membrane. Absorption of light in the form of photons. This absorption happens where? By light harvesting complexes, pigments that are present in the photosystems, that is purely thylakoid lamellae. And here photolysis of water, which is resulting in the formation of 2 protons, 2 electrons and oxygen atom. This is important. So this photolysis of water will happen where? Inside the thylakoid lumen. Okay, photolysis of water happens where? Inside the thylakoid lumen. So here immediately after absorption of light, this accessory pigments are submitting the energy to the reaction center, but this continuous supply of electrons uninterruptedly is happening because of the photolysis of water. The electrons which are released into the lumen, these electrons will behave or these electrons will act as a continuous supply to this primary acceptor, that is chlorophyll A. Do remember that. That is the main purpose of photolysis of water. And how that electron flow will be there, that we are going to learn under Z scheme now itself. So photolysis of water will result in the formation of electrons. These electrons will act as the continuous supply of electrons from the antenna or accessory pigments to the reaction center, and this photolysis of water happens where? Beta, in the thylakoid lumen. Clear, right? Next, assimilatory power. Assimilatory power, we have both ATP and NADPH. This ATP and NADPH are utilized for the reduction or fixation of carbon dioxide into what? Glucose. This fixation we learn in different cycles. What are they? What are the different cycles? C3 cycle, C4 and CAM, based on different plants. Okay, we have different cycles, we will be learning it in the light independent reactions. Today let us understand the formation of assimilatory power now, how ATP and NADPH are formed. We understood how photons are absorbed in the thylakoid lamellae by light harvesting complexes which are present inside the photosystems. Photosystems are present over the thylakoid membrane. This we have understood. Second is what? Photolysis of water. This is inside the thylakoid lumen. Okay, photolysis of water inside thylakoid lumen, where it forms protons, electrons and oxygen. This electrons will act as continuous supply of electron flow between accessory pigment and the reaction center. These two areas are clear for us. Now let us look into the formation of assimilatory power, that is ATP and NADPH. This formation of assimilatory power can be well read as phosphorylation, or this is what? Phosphorylation. So phosphorylation can happen in, phosphorylation happens in both two organelles, mitochondria and chloroplast. Fine, beta. This happens both in mitochondria and chloroplast. That is nothing but addition of phosphate group, ADP and inorganic phosphate. This addition of phosphate group to ADP, this results in the formation of what? ATP. ATP is formed both in mitochondria and chloroplast. But in chloroplast, that ATP is formed because of what energy? Light energy, that is photons. So that is why here it is termed as what? Photophosphorylation. Here it is termed as what, ma? Photophosphorylation. Phosphorylation. And here in mitochondria, it is not because of the light energy. So this is called as oxidative phosphorylation. Oxidative phosphorylation. Clear till here? Okay, so phosphorylation occurs. Very good morning, Deepak. Continue with the class. So phosphorylation occurs both in the mitochondria and chloroplast. Chloroplast, as light energy is involved in the addition of phosphate group, we are calling it as photophosphorylation, and in mitochondria it is oxidative phosphorylation. Now let us clearly understand this photophosphorylation. Photophosphorylation, if you have to look into beta, photophosphorylation. So here if you can see, two types of photophosphorylations are possible. One is cyclic photophosphorylation, cyclic photophosphorylation, phosphorylation, and the other one is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So here cyclic photophosphorylation will occur in bacterial photosynthesis or anoxygenic photosynthesis. Remember that. Cyclic photophosphorylation occurs where ma? In bacterial or anoxygenic photosynthesis. Anoxygenic photosynthesis. Fine. Wherein only stroma lamella is involved. Okay, only stroma lamella is there, and in the stroma lamella only one PS1, photosystem 1 is only present. When there is only photosystem 1, what will be the maximum wavelength here beta? P700. Okay, that is the maximum absorption range or wavelength of what? Reaction center. So here cyclic photophosphorylation occurs where ma? In bacterial or anoxygenic photosynthesis. Here only stroma lamella is involved, which has photosystem 1, that is P700. Clear, right? So this is important. They may ask us the questions regarding the organelle or the questions regarding cyclic photophosphorylation occurs in bacterial or anoxygenic photosynthesis or not, or they can ask us what is the maximum wavelength of light. Next, non-cyclic photophosphorylation. This occurs in higher plants, okay, where in oxygenic photosynthesis. In oxygenic photosynthesis, and this occurs in the higher plants in thylakoid lamellae. Okay, in thylakoid lamellae or thylakoid membrane, where two photosystems are involved, that is PS1 and PS2. Two photosystems are involved over here. Clear, right now? So now let us learn both in detail, cyclic photophosphorylation and non-cyclic photophosphorylation. First, let us look into the cyclic photophosphorylation. Here you can clearly see, as I told you, what light is absorbed in the form of photons, and this happens, photosystem 1 is present only in the stroma lamellae. Stroma lamellae is involved over here. Fine. So here you can see the reaction center maximum wavelength is what? P700, that is chlorophyll A. Maximum wavelength is P700, and the electrons are submitted to FRS complex. Okay, ferredoxin reducing synthase complex, FRS complex, it is submitted, and the primary acceptor is what? Ferredoxin. What is the primary electron acceptor in the cyclic photophosphorylation? Then what should be your answer? Ferredoxin. Okay. Next, this then submits the electrons to what? Cytochrome B6, then cytochrome F, plastocyanin, and it gets back to the photosystem again. Electrons will come back to the photosystem again. So here only ATP is formed. Only ATP formation takes place. There is no NADPH formation. Only ATP is formed in cyclic photophosphorylation. That also it is here only. ATP is formed. There is no NADPH that is formed over here. No NADPH formation occurs here. So here absorption of light in the form of photons, and then this electron energy is submitted to ferredoxin, which is the primary electron acceptor, cytochrome B6, cytochrome F, plastocyanin, and then electrons will come back to what? Photosystem. This is what ma? Stroma lamellae. Occurs in stroma lamellae, cyclic photophosphorylation. Only ATP is formed. There is no involvement of NADPH. Maximum wavelength is P700. Occurs in stroma lamellae. Remember all these points. This is very important. Okay, and most importantly, there is no photolysis of water. There is no photolysis of water. Photolysis of water occurs in non-cyclic photophosphorylation only, because in non-cyclic photophosphorylation we have photosystem 2. Here there is no photosystem 2, here we have only photosystem 1. Clear with this ma? Cyclic photophosphorylation. Now let us look into the non-cyclic photophosphorylation. First let me explain you the non-cyclic photophosphorylation in detail, then we will go with the diagrammatic view. See ma, assume this as what? Thylakoid membrane again. Let me take this small area over here, and you see here. Okay. So, this part is what? Stroma, and this layer will be what? Thylakoid lumen, and this is thylakoid membrane. Remember that. Okay, I have taken this small area over here, remember this. And here photosystem 2 is placed prior to photosystem 1, not because of its number, okay, but then because of their discovery. First PS2 is discovered. So, this is what? PS2, photosystem 2, and here we can assume like photosystem 1, okay, photosystem 2 and photosystem 1. So, here you see ma, firstly absorption of light in the form of photons happens by light harvesting complex, accessory pigments, and then in thylakoid lumen as a result of this, here we have one complex. That complex name is what? Oxygen evolving complex, OCE, oxygen evolving complex. This is adjoined to what? Photosystem 2, and this results in photolysis of water, where water molecule is photolysed to protons, electrons and oxygen atom. This electrons will serve as what? Continuous supply to the reaction center, okay, continuous supply to the reaction center. Let me draw it over here, okay. This is photosystem 2, maximum wavelength is what? P680 nanometers, P680, that reaction center which is chlorophyll A. From here primarily the electrons are accepted by what? 2 electrons are accepted by pheophytin. What is the primary electron acceptor of non-cyclic photophosphorylation? If that is the question, what should be your answer ma? Pheophytin is the primary electron acceptor. Next is what? Plastoquinone, okay. This plastoquinone will be taking the electrons, okay, plastoquinone, as it is taking the electrons from the pheophytin, it parallelly accepts protons from the stroma. Remember that, okay. Protons are taken from the stroma and parallelly it is taking the electrons. That is why it will be converted to plastohydroquinone, that is PQH2, okay. It is converted to what beta? Plastohydroquinone, and then protons are submitted to thylakoid lumen, and electrons will be submitted to what? Cytochrome B6F, okay. Cytochrome B6F complex, electrons will be submitted. Electrons will be submitted to cytochrome B6F complex, and then to plastocyanin. What is the ultimate electron acceptor of photosystem 2? If that is the question, the ultimate electron acceptor is what? Plastocyanin. From this, 2 electrons are being given to what? Photosystem 1. And here maximum absorption level of chlorophyll A is what? Maximum absorption level of chlorophyll A is P700. This is the maximum absorption range of chlorophyll A. From this plastocyanin, the primary acceptor will again be FRS, that is ferredoxin. Ferredoxin, which is the primary acceptor in photosystem 1, and then ferrous sulfur complex. The ultimate electron acceptor is adjoined to the membrane of this photosystem 2. The last ultimate electron acceptor is what? NAD+, NADP+. This is towards the stroma of the chloroplast, okay. This is present where ma? In the stroma. Till ferrous sulfur complex, all the protein molecules which are involved in this translocation are present in thylakoid membrane, but NAD+ is present in stroma, okay. I will tell you why when I am explaining you the dark reactions. So, NAD+ location is what beta? Stroma. That is very important, remember. This NAD+, NADP+ is converted to what? NADPH, NADPH plus H+, okay, NADPH plus H+. So, here you look into photosystem 2 and photosystem 1. Let us recall everything more quickly. So, the 3 steps of light reaction. First is absorption of light in the form of photons that are being absorbed by the light harvesting complex which is present in the thylakoid membrane, and then Photolysis of water happens inside the thylakoid lumen by oxygen evolving complex, which is adjoined to the inner membrane of the thylakoid. Okay, inner thylakoid membrane towards the lumen. After this photolysis of water, two electrons which are formed as a product of photolysis of water will serve as a continuous supply of electrons to the reaction center of photosystem 2. From here, the primary electron acceptor will be pheophytin, and then it is plastoquinone. Plastoquinone accepting electrons from pheophytin, parallelly it takes protons also from the stroma, and this turns out to be plastohydroquinone, and it submits the electrons to cytochrome B6F and protons to what? Thylakoid lumen. As such, the protons from stroma are being kept into what? Thylakoid lumen. And here 2 electrons from cytochrome B6F are given to what? Plastocyanin. Plastocyanin is the ultimate or final electron acceptor of photosystem 2. From here, the electrons are being delivered. Electrons are again given to what? FRS complex, and the primary electron acceptor of PS1 will be ferredoxin, and then ferrous sulfur complex. Ultimate or final electron acceptor of this non-cyclic photophosphorylation. Non-cyclic photophosphorylation the ultimate electron acceptor of this non-cyclic photophosphorylation will be NADP plus. Where is NADP plus present? In the stroma, whereas all these electron carriers, those are protein molecules, these are present in the thylakoid membrane, whereas NADP plus is present in the stroma of the chloroplast. And this NADP plus will turns out to be what? NADPH plus H plus inside the stroma itself. So, this is all about non-cyclic photophosphorylation. If you have to see the flow of electrons, that is looking like a Z. So, it is called as Z scheme, and here more we can expect a questionnaire based on the uphill and downhill movement of electrons beta. So, here first uphill movement where you see from oxygen evolving complex or the accessory pigments, the electrons are being given to what? Chlorophyll A, that is reaction center. This is uphill, and then downhill towards the plastocyanin, which is the last electron acceptor of photosystem 2. Again uphill movement towards the P700 complex, that is photosystem 1. Again downhill movement up to what? NADP plus, that is stroma. Clear till here ma? Okay, uphill and downhill movement. And Z scheme, where are these protein molecules? Those are electron carriers present in the inside the thylakoid membrane. Ultimate or final electron acceptor is present in the stroma of the chloroplast. Clear with this beta? Non-cyclic photophosphorylation or Z scheme. Fine ma? Enough. Here you see this complete picture. When diagram based questions are given also, you should not we should not escape our score. So, here you can see, right, oxygen evolving complex, this is in the thylakoid lumen, which forms out to be oxygen and protons, electrons are present, and these electrons are submitted to what? The photosystem 2. Plastoquinone, can you observe here beta? Plastoquinone is submitting the protons from stroma to the lumen. Fine. Cytochrome B6 complex, plastocyanin is located towards the inner side of the thylakoid membrane. Look at here carefully. Plastoquinone or pheophytin is present over the outer side of the thylakoid membrane. Plastocyanin towards the inner side of the thylakoid membrane. From here, plastocyanin are given to what? Photosystem 1, where the primary acceptor ferredoxin, ferrous sulfur complex, and NAD plus, which is present in the stroma. Here NAD plus along with hydrogen, it is getting reduced to what? NADPH. This gets reduced to what beta? NADPH. And here the enzyme which is involved in this reduction process of NADP plus to NADPH plus H plus is nothing. The enzyme which is involved is what? NADP reductase. NADP reductase is the enzyme which is involved over here, that is reducing the NADP plus to NADPH plus H+. Till here it is clear na beta, and how ATP is formed, that we are going to look under the chemiosmotic hypothesis. So whatever we have discussed today regarding the cyclic and non-cyclic photophosphorylation is clear to you, Nama. So here you can clearly distinguish between the cyclic and non-cyclic photophosphorylation, where in cyclic photophosphorylation only photosystem 1 is involved. Here both the photosystems are involved, PS2 and PS1, and here there is no oxygen evolving complex, there is no photolysis of water in cyclic photophosphorylation, whereas in the Z scheme you can see the oxygen evolving complex and photolysis of water, and here you can see both ATP and NADPH formation in the non-cyclic photophosphorylation, but here in the cyclic you can see only ATP formation, and here the electron movement is in a cyclic manner, but here in the Z shape. So it is called as what? Z scheme as well. Fine, ma. So these are the primary differences. Here only stroma lamella is involved, here thylakoid membrane it occurs, and here only here PS1, here PS1 and PS2. So this is all about cyclic and non-cyclic photophosphorylation. Let us exercise the differences more keenly. So here if you see, first cyclic photophosphorylation, synthesis of ATP, okay, synthesis of ATP during the light reaction of photosynthesis by cyclic passage of electrons, and from P700 when plants need more ATP than NADPH. Okay, so this happens when they need more ATP than NADPH. Here synthesis of ATP during light reaction of photosynthesis, in which the electron donor is required and oxygen is produced as a byproduct. Here there is no oxygen evolved. This occurs in isolated chloroplasts and in photosynthetic bacteria, as we have discussed in anoxygenic photosynthesis. But non-cyclic photophosphorylation, this will occur in higher plants, algae and cyanobacteria. And here if you see, ma, this cyclic photophosphorylation occurs in anoxygenic photosynthesis, where there is no oxygen evolved. But here this occurs in oxygenic photosynthesis, where the oxygen is evolved. This final electron acceptor is P700. Here final electron acceptor is NADP+. Few more differences to be revised. Electrons move in a cyclic manner in cyclic photophosphorylation. This in a line or a pattern of what? Z scheme. Okay, this line or a pattern, this move, electron flow is in the pattern of Z scheme. Only photosystem 1 is involved over here. Here both photosystem 1 and photosystem 2 will be involved in non-cyclic photophosphorylation. Electrons are first expelled, from the reaction center of PS1, okay, but here electrons are first expelled from the reaction center of PS2. Here you see, ma, electrons return to the P700 after passing on the electron transport system. Electrons will return to P680 complex over here after photolysis of water, and this is accepted by the photosystem 2. Clear, right, ma? With this we have finished the 3 sequential events that are happening in the light reaction. What are they? The absorption of light in the form of photons, photolysis of water and assimilatory power formation. In that, today we have seen how NADPH is formed in the non-cyclic photophosphorylation. How ATP is formed we will discuss in tomorrow's class. ATP formation through what phenomena? That is chemiosmotic hypothesis. Through chemiosmotic hypothesis, how ATP is being synthesized, we will be discussing in tomorrow's class. Okay, and then we will also begin with dark reactions in tomorrow's class. Okay, and I would like to wind up this session by giving you a brief gist on NEET World's special programs. Here in the NEET World, we have NEET World's 2 steps to medicine for 11th standard students and NEET World's 1 step to medicine for 12th standard students and NEET World's power step to medicine for the repeaters. For any of these 3 programs, the fee is very minimal beta. It is just 3333 per month, which is highly useful to all the students of different financial backgrounds. So make use of these programs, wherein this minimal fee only you will get the complete lecture content with 2 master revision programs and complete study materials also. The course is available both in offline and online mode. To know more about this, you can directly contact the given number, or you can visit our branch which is in Narayanguda, Hyderabad. To log into our Telegram group is highly beneficial to all the students who want to study in the public platform or who want to crack NEET 2026. For there, who want to crack the NEET 2026, just a second. NEET 2026 through self-study. To all the students who are opting this platform, this will be the best choice to join the NEET World's Telegram group, wherein you will be allotted with daily quizzes of botany, chemistry, physics and maths. Daily MCQs will be provided and live class schedule is provided in this Telegram group. Free resources of study materials might be, or practice questions might be, are given in this, and one-on-one doubt solving session with the faculty of NEET World are possible with this Telegram group. To join this Telegram group, you can directly scan this QR code, or you can join this group by our official website, which is www.neetworld.in, or you can directly call us onto the given number. Fine. So make use of all these programs and don't miss this session of 100 concepts for NEET 2026 for all the subjects that we have launched here. Each and every class will be helpful for you, and most importantly, highest beneficial class is your questionnaire session. 1000 questionnaire session series which are there, that is 100% helpful for you beta, where we are going to exercise the maximum amount of questions for you people. Okay. So thank you for today and we will meet tomorrow.